Let's record. Okay, we're recording now. So welcome back from Thanksgiving holiday. So today is the second to last lecture. So we'll go over some more sort of advanced recon today. And then on Wednesday, we'll sort of sum up the course and then also talk about projects and sort of remaining logistics for the course. So just 2 lectures left. Before we begin today, are there any questions about projects, course logistics, or anything else about the course? Okay, so in terms of remaining deliverables, I think the only thing remaining is homework 7, right, which has just got 2 problems on it. Second one is just mostly just going through and trying some stuff out. So hopefully that'll be okay while you're working on projects. Other than that, I don't think that there's anything else aside from your project that's sort of going to be coming up for the class. All right. So if there are no questions, let's begin. So I did want to review a little bit about T1 and T2 contrast, simply because on one of the questions there was the T1 weighted question. There was some sort of diversity of answers, and so I just wanted to make sure everyone was on the same page on that. So if we have 2 tissues with different T1s, which is shown here. Let's see if we can get this mouse going. Hello. Okay, there we go. So these are the longitudinal recovery curves for 2 tissues with 2 different T1s, right? And if we look at the difference between the 2, the peak is around 1,000, which is roughly the average of the 2 T1s. Right. And so we said that a good rule of thumb is if you have 2 tissues with different T1s and you want to sort of maximize the difference between the 2, then you should probably just pick the average of the 2. So then we can do the thought experiment. Let's say I took one of these ones. Let's say we took the 1200 millisecond one and doubled it, right? So it'd go from 800 to 2400. Then the average is going to go up too, right? So that means that this curve, instead of being maximized around 1,000, is going to be probably maximized around, let's see, 2,400, around 1,600 or so. So that's going to, if the T1, the average T1 between your 2 tissues increases, then that's going to mean you need a larger TR. Okay, so that's the way we would think about that problem. Now, obviously, you weren't given a lot of information in the quiz problem, and so the question is, should you increase TE or decrease TE? And there it's sort of, you have to think about it a little bit, but it's basically the idea is because we're changing T1, then clearly we're more interested in T1 weighted contrast, okay? And so therefore, we don't want anything to sort of screw up with that. So if we increase TE too much, obviously at some point the signal would go away, right? So it's sort of ambiguous, and that would get rid of all our T1 contrast too, because we have no signal. So to be on the safe side, we would, if we had to make a choice, we'd rather decrease TE. So at least maybe we lose some T2 contrast, but we've got our T1 contrast. So that's very much like problems you deal with in everyday life, where either in research or in industry, you just don't have enough information, right? You have about 60% of the information you need to make a decision, and the rest you sort of have to sort of think, well, what would be the reasonable thing to do? So that's similar to that. Most people got this one right, but just to go over it, just to make sure that everyone's on the same page. In fMRI, what really causes the contrast are these iron atoms, right? And so the more iron that's exposed, the more they're going to perturb the magnetic environment, and the more they're going to cause dephasing, and therefore increase the rate, right? And so that's shown here, where if we have more, and the more iron means more deoxyhemoglobin, because deoxy means we've taken the oxygen off the hemoglobin, okay? So that might just be a sort of a terminology thing, where oxyhemoglobin is when you've got oxygen on the hemoglobin, and deoxy is when you take it off. And so more deoxyhemoglobin, you can think of it as just meaning more iron. Right? And so that once you have that picture in mind, then it should be pretty straightforward. And in fact, I think if you this was essentially gave you sort of the response you needed. Right. Okay, any questions on those concepts before we move on to today's lecture? So what I want to go over today is, and this will sort of walk you through problem 2 of the homework and sort of point out what you should be looking for. And for problem 2, you just have to make a very minor modification and then just sort of report on what you see. So we're going to review a little bit about what we talked about with image reconstruction. to get into what's called parallel imaging. I think there's 3 or 4 projects on compressed sensing, so we'll talk about that a little bit. Deep learning has revolutionized medical imaging over the last few years, and so we definitely need to touch upon that. I doubt we'll have time to do this, but I just put that in there just in case we went through the other material, but we probably won't get to that. So on Wednesday, we'll finish up with some other really interesting applications of MRI. Okay, so just to remind you that throughout this whole course, we've been talking about going between image space and Fourier space because we collect the data in Fourier space or K space. And the reason we do that is just because we're able to manipulate the phase of the spins, right? And so that allows us to do the Fourier transform in a very straightforward fashion. And so we talked about in a previous lecture, we said, well, we can think of this as a 2D image, but you can always take anything that's 2D, and if I go column by column by column, I could make this into a long vector. And then I can also make my signal into a long vector. And I can just represent the relationship between the two by multiplication by these x composite exponentials. Right? And so once we have it in this format, then we know we're in the realm of linear algebra, right? We just have a system of equations, right? And so it turns out that when we look at either parallel imaging, compressed sensing, and deep learning, this is sort of the framework we want to be in, okay? Because all of those are going beyond Fourier transforms. Okay? So Fourier transforms was the bread and butter till about 15 or 20 years ago. So that's all you needed to know for medical imaging, aside from some sort of very rarely used techniques that people came up with. Now, in the last 15 or 20 years, especially with the advent of more computing power, now that's not sufficient, and the next generation is going to have to be more familiar with sort of looking at things as sort of linear equations. Okay, so here's a picture of, we typically think things as in 2D, but we can take this 2D image and vectorize it. And then this 2D sinc function is the Fourier transform, and that can also be vectorized. And it turns out this vector equals this vector multiplied by this encoding matrix, which just has a bunch of all those complex exponentials laid out in it. All right. And so in your homework, you sort of did that, and you played around, you sort of just looked at the properties of this encoding matrix, and you found out that all the columns of these are orthonormal. Okay, that's a very special property, right? If you take a column with itself, give you something, but a column with any other column is going to give you 0, right? So this is a special property of the Fourier transform matrix, which is it's orthonormal. It has orthogonal basis functions. If you go back to linear algebra, they're all perpendicular to each other. Now, it turns out that once we go beyond Fourier transform, or if we go to, for example, there are other encoding schemes where that may not be the case, but for now, we'll assume that these are largely orthonormal. So what we've been doing most of this class is treating the case where we want to have this to be a square matrix, right? The number of equations equals the number of unknowns. That's the Fourier transform. So if I have like 128 by 128 image, then I'm going to have 128 by 128 points in K space. So sort of the same number of points in both space. And that basically, because this is orthogonal, the inverse of this matrix is transpose. Okay, so it's a square matrix, so I can always think about taking the inverse, but I can also just take its what's called conjugate transpose. Right? And that's because the property of this matrix is orthogonal. Right? What we're going to talk about today is 2 special cases, and we talked about this a little bit before, which is one case where we actually have more equations than unknowns. Okay, and in that case, as you know from linear algebra, there may not be a solution, right? The solution may lie outside of your model space. So we want to talk about what to do there. And then we also have underdetermined, where in this case, there are multiple solutions. Okay, and this is what happens with aliasing. Okay, when you don't have enough data, let's say you don't have acquire enough data in K space, then there's actually multiple solutions that are possible. It just turns out the alias solution that we found is what's called the minimum norm solution. Okay, it's one of many solutions. But what we'd like to do is we like to do underdetermined because obviously we don't have to acquire as much in K-space, so we can go faster. But we don't want the imaging artifacts. And so today's class is all about how do you go faster than you think you can? How do you break the Nyquist limit so you can go faster but not have artifacts? Right. So that's today is sort of breaking the speed limit. This is just reminding us that if this is square, then the inverse of that is just the E conjugate, and then that's sort of in MATLAB is just ENC to this transpose notation. So MATLAB knows if it's complex to take the conjugate, so that's very easily done. Okay. We could also do what's called the pseudo inverse. So in this case, we don't have to do pseudo inverse because it's square matrix, but the pseudo inverse is what we'll typically, if you look through the code, we'll use this a lot. So the pseudo inverse works if you have either a square matrix or a rectangular matrix. It doesn't really care what the shape of the matrix is. There's always going to be a pseudo inverse that MATLAB can find. Right. So the first question is, how do we get overdetermined equations in MRI? And the answer is you have lots of coils. Okay. So really you only need one coil. And for up until about 20 years ago, if you wanted an MRI scanner, there would just be a single coil. Okay, so for the first 20 years of MRI, basically, mostly people were just working with one coil for transmitting, another coil for receiving, and just one receiver coil. Okay. Then about 20 years ago, people started playing around with multiple coils, and then they realized they could use these multiple coils to go faster. And remarkably, that innovation was done by two graduate students, one in Switzerland, one in Boston, and they came up with different methods. And so the bread and butter of what we see today with parallel imaging was basically invented by people like you. Okay. So the idea is we have multiple coils, and each coil is going to see a different part of the brain. Right? And that's key because realize if you want to make an image, if all the coils see the same thing, then you're not really going to be able to speed things up, right? Because they're not adding any information. But for example, this coil sees this and this coil sees that, then that might give you enough information that you're like, well, these coils are telling me about different parts of the image, so I don't need to encode everything. I don't have to spend all my time encoding that. I should be able to use these different coils to help me go faster. So we'll walk through this. This is what it looks like. So basically, instead of just one encoding matrix, so before we just had let's say we just had one coil, and we have a square matrix, and then it's multiplying the object. Now you see we have coil 1 sees this, coil 2 sees something else, and coil 3 sees something else. Okay. They all have the same encoding matrix, okay? And the F just means the full encoding. Now the only difference is they each have a different coil sensitivity. Okay, so basically if you look at, for example, this is my object, and these are coils at different locations. You see the image from each coil is slightly different, right? It's brightest close to the coil, and then it falls off. Okay, so each coil sees something differently. And those differences can be represented in a diagonal matrix with coil sensitivities. Essentially, we take our object and multiply it by the coil sensitivities. Okay. So for example, this coil sensitivity is pretty bright here, close to the coil, and falls off here, whereas this one's bright on this side of the object and falls off over here. All right. And so now what we've done is we've created an overdetermined system. Right? We have more equations than we have unknowns. And so we don't necessarily have a solution that's going to give us the exact, that's going to minimize, make the error go to 0, but we can pick the solution that's the best solution. Just a little bit in terms of your homework. So in the homework, we defined some coil profiles, which we'll look at in a minute. And so these are some, this is like one coil profile. So what you're going to be asked to do in the homework is just come up with your own coil profiles and play around with them. Okay? And that's really for problem 2, that's all you need to do. And the rest of the code you can pretty much leave alone. Right? So literally 1 or 2 lines of code changes in your homework. Just to remind you, when we have an overdetermined system, that means what we measure, which is some vector out here, may not be explained by our basis functions or our model functions. And so we're going to always have some error. And the job of the least squares estimate is to minimize this error. Okay? We want the whatever estimate this is the best estimate we come up with, and that estimate should be closest to my data as possible. And this error will be minimized when the error is perpendicular to my estimate, okay? Or the models. This whole model space is the plane of possible solutions. So if I picked any other solution, for example, if I picked, a solution like this, then this would have a much bigger error. So it's the perpendicular is going to have the smallest error. And so if you do that, then you end up with this very sort of well-known expression. This is sort of the least squares estimate if you have an overdetermined system. And this whole thing is essentially what MATLAB gives you as pseudo inverse. Sorry, this is a busy slide, so let's walk through it a little bit. The whole point of this slide is just to tell you a little bit about what approximation we're going to look at. So if I have an overdetermined system, then I can always write the solution like we just did. This is the least squares estimate here. Okay. Now it turns out if I write this out in terms of all the E's and F's and C's, I get that the estimate is essentially equal to, if I did each image individually, okay, I could take, so each coil sees, each of these coils, since it's a fully sampled system, I can come up with an estimate of the object, right? Because I have enough case-based data, so each one is not aliased, right? And now the question is, how do I combine all these to make my estimate? So it turns out that the solution has this form, okay? So you see each of these is individual image. It's multiplied by something to do with its coil, and then it's divided by all these sums of coils estimates. So we don't necessarily know what the coil profile is. I mean, we could measure it, but if you didn't want to measure it, turns out a very commonly used approximation is the coil sensitivity is just the value of this image, okay, divided by the square root of sum of squares of all the images. Okay, saying that if this is, let's say this is the sum of squares of all the images, then square root of sum of the squares, then this coil sensitivity is, the sensitivity of this coil is pretty much this guy divided by this guy. And it turns out if you do that, you end up with a very simple but widely used approximation for how to combine these coils images. It says take all these images, square each one, sum them all up, and then take the square root. Okay, so that's known as square root of sum of squares, or SOS reconstruction. And so you'll be asked on the homework to sort of compare that to the ideal. And let's look at some examples of that. So here's an example here where, for example, let's say coil 1 sees this. So obviously the coil is on one side of the frame, and the coil 2 sees the other side of the frame, right? And it looks like if I took the sum of the squares of these two, I get a pretty good looking image. If I take this guy and divide by this, then that's my coil estimate. That's my sensitivity estimate. So you notice it's brighter here and darker here. If I take this and divide by this, then this is brighter here. And so this is my estimate of the coil sensitivity. Right. It's a little noisy, so in practice you might smooth it out a little bit with some filters. But essentially that's what the sum of squares is doing. It's estimating the coil sensitivity by this function here. Right. So I'm going to walk through this fairly slowly because this is sort of what you're going to see when you do. This is like what figure 3 is going to spit out. Yes, question.
So this is if there were artifacts in either of coil 1 or coil 2, would that also get squared and added up in time?
Yeah. So that's why you would typically low pass filter it, because you're trying to get rid of, like even here you're sort of seeing there's a lot of stuff out here, and so it's not perfect, but then typically if you have artifacts, they'll be in all the coils, and so you're no worse off than if you just have one coil. Yeah. If it's due to the sampling. Okay, so this is sort of, we're going to walk through a couple of slides like this. So this is what you're going to see when you do figure 3. And so these are the two coil profiles. So we're going to start off with two coils. One where, so this coil falls off very gently this way. So it's more sensitive here than here. And here where it's more sensitive here and falls off. But these coils basically see the same thing. right? So they're not very diverse, right? And so these are two coil profiles. You'll be asked to come up with your own coil profiles. We'll look at two coil profiles today in class, and you're just use your creativity. You can come up with whatever you coil profile. And the comment in the homework is it doesn't have to be symmetric. You could have one that only spans this and the other that spans that, or do whatever you want to see what you get, okay? There's no right or wrong answer. It's just for you to experiment with it. This basically shows you, and so we're going to assume this coil profile varies in the y direction, so along rows. And so here it's bright here and dark here, here it's dark here and bright here. Okay, so that's just what the coil profiles look like, and they're only varying in the y direction. So if I take coil 1 and multiply by my object, which is a uniform square, I get this, and otherwise I get this. And here in this case, I've added some noise to it. Okay. Or not yet. I haven't added noise to it yet, but this is what the objects look like. So they look, basically the objects look almost the same. And then here is if I do the recon, the least squares recon, and I've added some noise to it. So in your homework, you'll look at it with and without noise. The slides here all have noise in them, and so you can sort of see I've added noise to the reconstruction. And so you can sort of see here, it's a little noisy, but it's pretty good. Okay. And if I look at the sum of squares recon, that's also pretty good. Okay. So when the coils have these very gently sloping profiles and largely overlap each other, sum of squares actually does a pretty good job. And so it used to be that when people were designing coil arrays, they were like, oh, well, if I'm going to use sum of squares, I want to make these coils have as much overlap as possible. Okay. And that was the name of the game. But that turned out to be exactly the wrong answer if you want to go faster, which we'll see in a minute. And so here you're seeing both coils, coil 1 reconstructed individually, this should say coil 2 reconstructed individually. So I think that's a mistake, definitely. All right. Now here I've got a different coil profile. So this is the second coil profile that's sort of default in the code. So it sort of here, this drops off like that, and this drops off like that. So you have two coils, each covering about half of the object. So this coil 1 is bright here and then falls off. This one is dark here and gets more. Okay. And so if you look at the object multiplied by the coil sensitivity, this one captures the top part of the object, this one captures the bottom part of the object. If I look at the reconstruction though, it's pretty good. I have enough data, I can stitch together a pretty good estimate. And then if I took each coil and reconstruct it on its own, and then take the sum of squares, I get this. And now you can sort of see it's not so good, right? There's some weird sort of shading here. Right? So in terms of if you want to do sum of squares, having things that don't overlap very well is not so good for your sum of squares recon. So that's, if you pick, for example, a coil profile like this, then you're going to see hopefully that sum of squares is not such a good estimate, depending on what your coil profile is. Obviously, you're not limited to just one coil profile. Feel free to experiment, but for the homework, you just have to do one additional coil profile. Okay, any questions on that? All right. So now we're going to talk about how to go faster. So far, we haven't really talked about going faster. Like we just said, we're going to add more coils. Now we have more data. What do we do with all that data? Well, one thing to do is take the square root of sum of squares. But we haven't gone faster. We're still collecting all case space. To go faster, we need to start skipping lines in case space, right? Because each line takes time. So we start off with fully sampled acquisition, and we can sort of go back and forth. If we do undersampled acquisition, let's say we skip every other line in KY, then we're missing all these lines, and so we know we're going to get an aliased image. Okay. And so what we'd like to do is somehow still go faster, but get rid of that aliasing. And so for parallel imaging, compressed sensing, and deep learning, the whole question is how can I go faster but not have aliasing? Turns out that this alias image is just equal to what's called the minimum norm estimate, which I just take this undersampled encoding matrix, take its transpose, and multiply by S, and that's basically the reconstruction. As we said last time, the minimum norm estimate is just that estimate that that sort of has the minimum norm. So all of these are possible solutions. and we're just picking the one that is the smallest. Okay, so that's all minimum warning means. Okay, so once again, let's review why we want to undersample. Obviously, we want to go faster, so we can reduce the time and cost of the acquisition. Right? Time is really important for certain applications. For example, if you're scanning little babies, you don't have much time because babies move. Okay, so if your scan takes 10 minutes, you're going to end up with just this blurry image, right? So what you'd love is like a 1 second image, right? But you know, if you can get a 1 or 2 image, maybe the baby will stay still for that amount of time. Obviously, other cases are where someone is critically ill. You need to get them in and out as soon as possible. Okay. Cost of acquisition. Okay. Basically, Medicaid will reimburse you for a certain amount for each type of scan. If you can do that scan quicker, you can run more subjects through. You know, your radiology business makes more money. Okay. So radiologists are incentivized to have things go faster. So there's two main. There's sort of the different approaches to dealing with undersampling are, one is we're going to use more coils, and that's called parallel imaging. So multiple coils in parallel. The other solution, and these can all be combined somewhat, are in terms of using prior information or assumptions. And we'll talk about what that means. But here we're not making really any assumptions. We're just going to collect more data. Here we're going to say, well, actually, if I know something about what I'm looking for, I should be able to go faster. Okay. It's sort of like this. If you lose your keys in your house and you have no idea where they are, you've got to look over the whole house, right? But if you sort of know, well, the last place I was my bedroom, then you just have to confine your search to your bedroom. And that search is going to be much faster than going over your whole house. So the same thing is going to be true with going faster. If I have some prior information, I can either use compressed sensing or deep learning to go faster and still get a good image. Okay, just to remind you, if I have full sampling, I could certainly get a good image. One way I could go faster is I could just collect the center of K-space, right? I just skip these lines. The problem with that is that I'm going to get lower resolution, right? Because I haven't acquired those higher frequencies. The other approach, obviously, is skipping lines, right? But that leads to aliasing. But it turns out we can deal with that. Okay, so this is a busy slide. I just want to get one point across for this. So basically, the signal from the ELF coil is just going to be my object. This is the Fourier transform, and all I've introduced here is this coil sensitivity pattern. Saying that now each coil, I don't see the object, I see the object multiplied by the coil sensitivity. Okay. So this will turn out to be a little more important later. And then typically when we want to speed things up, we want to speed up like if we're doing spin work, we want to speed up the phasing codirection. Because we can zoom across K-space pretty fast in the readout. That may take a millisecond or two. So there's really not much gain in speeding that up. Okay. Whereas the phasing code requires line by line by line. So that's typically the speed up. So we'll typically do full Fourier in the readout. And so that means we can just do the Fourier transform along the readout direction. And that allows us to come up with what's called a hybrid space image, which is partly still in KY and now in X coordinates. Okay. And so now I basically have this thing where I have this sort of strange function here, which at every X prime location, it's still a function of KY, but I basically can solve for my object column by column now. Okay. So that just allows me to solve a lot of little problems. And that's what we're going to do. So essentially, we do the inverse transform along the readout direction, and then now we can just solve each column separately. And so this is now just a 1D Fourier transform that we have to solve. Okay, so let's take a look at this. So it turns out that, as we mentioned last time, the way we get around it with parallel imaging is we take this undersampled system and make it oversampled, or overdetermined. So this is underdetermined, right? More unknowns and equations. Now we get more coils, right? We've got 1, 2, 3 coils now. So now we are overdetermined. And we should be able to now get an estimate that doesn't have aliasing in it. So we're using the information from multiple coils to undo the aliasing. It turns out that the solution, these matrices can be pretty big, okay? And so if you're going to actually if you just put in your homework, they're relatively small matrix sequence. So, but even with MATLAB, you'll notice it sometimes takes a little, like 10 seconds, which is long these days, right? You have to wait 10 seconds for something, it's MATLAB's really thinking. But if it was a really big matrix, it would just take a long time. Okay, and it may take longer, unless you had a GPU or something, it may take too long for your clinical system. So ideally, when you scan someone, you want the images right away so you can look at them. You don't want to wait an hour or something. So let me just walk you through the approach that was come up to solve this, and realize you can actually solve this as a bunch of really easy problems. So imagine this is my object here, right? And these 2 points in my object, if I undersample by 2, they're going to be aliased, right? Because they're half FOB apart, right? So if I undersample by 2, I'm going to have replicas every FOB over 2, right? So I can look at these 2 things that are going to be FOB over 2 apart. Okay, these objects, this one is multiplied by this coil sensitivity, and this one is multiplied by this coil sensitivity. Okay, and so I can imagine that in my image, if I had fully sampled, this would give me A and this would give me B. All right. Now I have 4 different coils. And now I'm undersampled such that these guys lie on top of each other. Okay. So the value of this voxel is actually A plus B for the first coil. So A1 plus B1, which is just equal to the original value I sub A times the coil sensitivity from the A coil plus IB times the sensitivity from the B coil. All right. So basically, each of these alias voxels is the sum of 2 voxels coming from different locations, but multiplied by different coil sensitivities. And because each coil sees something differently, you notice the only thing changing in these equations is those coil sensitivity patterns. Right now, I've got coil 2, I've got coil 3, I've got coil 4. So I can write each of these as what I measure, F1, F2, F3, F4, this is for every voxel, is equal to what I want to estimate, these 2 unaliased, right? So F1 is the aliased version, I and IB are the unaliased version, and I can relate these by these coil sensitivity patterns. This is just a general linear, this is just an overdetermined system. So I should be able to solve this. If I measure these, I can come up with that. All right? So that's what's called sense recon. So that's sort of walking through it from just sort of, how you might think about how it's sort of convincing you that even if 2 voxels land on the same place in your image, if you have enough information, if you look at them from the different coils, you can undo that aliasing and split them back into what the original values were. And you just do that voxel by voxel by voxel. Each of these is a very small problem, so you can do them very quickly. Okay. Sorry, this is a really busy slide. The whole point of this slide is just showing you that if you go through the actual equations, okay, then it turns out you end up with the thing you're trying to solve is you're trying to take the inverse of something that's block diagonal. Okay. And because it's block diagonal, the inverse will also be, you just have to take the inverse of each one of these little blocks. And that basically is another way of realizing you can break down this big matrix into like little blocks, which is exactly what we just did on the previous slide. Okay. So if you go through the math, you end up with exactly the same equation. But just showing you, you can go from the theoretical point of view, get exactly the same equation as just talking through it, but now at least you have a rigorous way of doing it. Okay. So once again, we end up with just this problem where we're trying to estimate these 2 unaliased values and by measuring different coils, and the solution is just here, okay, where we basically, this is the pseudo inverse, and we multiply our measured data from every coil, and this is the coil sensitivities here. Okay, so that's known as sense recon, and that is actually still used a lot on scanners. It varies a little bit on which scanner you use. For example, if you use a Philips scanner, that will always tend to use something called sense. Okay. If you use a G scanner, it sort of varies. You can do something similar to Sense, but there's another method which we'll talk about in a little bit, which is called GRAPPA, which is used a little more on, for example, like Siemens scanners. But they all have the same principle. Now, the next thing we want to talk about is what happens with these coil. Like if you want to do this, what should be the optimal coil profile you want? Okay. So we said before, if you're doing overdetermined, then the sum of squares works really well. These coil profiles are relatively changing very slowly, so the coils have a lot of overlap. But that was not, and then if the coils didn't overlap so much, then that was not so good for a sum of squares. Now, if we're trying to go faster, you would think that probably the answer is going to be a little different, right? Because if all the coils see the same thing, it's going to be hard to go faster, right? But if each coil sees something a little different, then that gives you more information about what's going on in the object. So that should work out better. Okay, and so the way we do that is we start looking at what's the covariance in my estimate. Okay, so I'm just going to talk you through those slides. Don't worry so much about the equations, just sort of realize that we are just talking about how noisy is my estimate going to be. Okay. So basically, at every voxel, right? You know, we have sort of different coils, and the coil, first of all, the noise on different coils may correlate because they sort of, if they're slightly overlapping, they're sort of seeing the same thing, so they might get the same noise. And if you introduce the covariance of that noise, then you just change your estimate to incorporate that noise term. Right? What's more important is you can actually calculate what the covariance of your estimate is, and it's given by this. It's this coil sensitivities, and then this whole thing inverted, and then this is the correlation of the coils. Now, it turns out if C is square and invertible, then this estimate doesn't really depend on the noise correlation. It's just, you're just going to invert that matrix. And so in that case, it doesn't really matter. But in most cases, we're not dealing with a square matrix, as we saw. So what we're going to introduce is something called the G factor. Okay. And the G factor is basically the SNR that I get when I go faster divided by the SNR I go when I go full Fourier. And we're just going to look at some plots that explain this G factor. So first of all, let's explain what R is. R is acceleration factor. So R is just how much faster you go. So like R is 2, it means you skip every other line. You go twice as fast. If R is 3, you go 3 times as fast. Okay. And so you can sort of see there's already an SNR penalty. We'll talk a little bit probably on Wednesday now. Basically, because you're collecting less data, your SNR will go down. And so that's something that you just have to live with. But in addition to that, there's something called the G factor, which is geometry factor. It's location-dependent noise amplification. That depends on how different the coil sensitivities are at that voxel. So let's take a look at an example of this. So these are 3 different images with different coil profiles. And so, for example, here, the coil spacing is, this image is acquired with coils that sort of overlap a lot. And then as the coils are further, as the spacing goes from 1 centimeter to 3 centimeters, 6 centimeters, as you make those coils overlap less, you notice the images get much better. Okay, this is got a lot of noise in it. And then this progressively gets better. And that is represented by what's called the G factor. So you can sort of see here in this good looking image, those G factors are very close to 1. Okay. Whereas here in these areas, you can sort of see they're close to 5. There's a lot of noise amplification here. So we're going to take a look at that in our homeworks. This is just to show that there is a formula for that. We're not going to go through it. And this is just showing you how that's calculated in the code. Just for the homework, all you just need to know is that, you know, you're just maybe plotting this G factor and looking at it. Okay, so let's take a look at this now. So this would be like figure 6 in the code. And so when you're playing around with it, you'll typically look at figure 3 and 6, and then maybe figure 7. So here are the coil profiles here. And now, if I reconstruct each image on its own, it's highly, you see they're both aliased. But if I calculate it with the pseudo inverse, I get a pretty good looking image. I mean, there's added noise, but that's okay. And if I look at these G factors, you can sort of see they're fairly close to 1. So I really haven't amplified the noise too much. That's good. So I have these coils that each see something quite different, but they don't overlap very much. And so my estimate looks pretty good. On the other hand, look at this one. Now I have these 2 coils here. And if I reconstruct each, they're both alias and they look pretty much the same. And now if I try to come up with my estimate, it looks really, really bad. Okay. So what looked pretty good if I had sum of squares now looks horrible, right? This took the same amount of time to acquire this. These images have taken the same amount of time, but just by changing that coil profile, I've gone from a relatively good looking image to a horrible image. Okay. And if you look at these G factors, the scale here is I'm up around 18. Okay, so I've basically amplified my noise by a factor of 18, just because I've selected my coil profile in a non-optimal fashion. So this is a big deal because this means that when people are designing these coils now, they really have to take this into account. So if you design a bad looking coil, if you design a coil that has really bad G factors, no one's going to pay you money for that. And these coils are very expensive. These coils can run anywhere from $50,000 to $100,000 for a coil. So if your coil costs $50,000 and gives images like this, you're not going to have a business. Okay, so these are the coil profiles, and these are the sort of G factors that you can sort of see. So these are around 1. This is like figure 7 from the homework from your MATLAB code, and this is like these are around the 18s. You notice they're both sort of, this is pretty close. They're basically 1 for these voxels, which would correspond to voxels here. That's because if I go half an FOP from here, this is 1, and it's kind of part of 0. So they're very different, right? But notice the G factor does go up a little bit here in the middle and also at the ends. So why does it go up here at the end? Well, look here. If I'm here and I go about half an FOV, then I'm here, right? So I'm getting 1 on one coil and maybe like a half on the other coil. So now the coil sensitivities aren't so different, right? So the more some of those sensitivity patterns are for that voxel, it will tend to amplify the noise a little bit. But it's still different enough such that it's only about 1.2, or that's much better than 18. So this is what you're going to be looking at in your homework. Just whatever coil profile you look at, just look at sort of its effect on images and also the G factor. All right. Any questions on that? Okay. We're just going to talk a little bit about grappa, and just to show you how the concepts from this course are actually used in the real world. You remember that if we have my object and I multiply by coil sensitivity pattern, I can get, this is my image, right? This is multiplication in the image domain. If I take the Fourier transform, it looks like this. And this means I'm convolving with the Fourier transform of my coil sensitivity pattern. So this is now convolution. And so that means I'm going to take my actual Fourier transform, I'm convolving it with this thing, and I'm going to end up with a smooth version of my Fourier transform. What that means is the data I get in Fourier space for that coil, this pattern is a smooth version of this, which means that I should be able to take advantage of that smoothness, right? If something is smooth, it means that if I know the points around it, I probably can estimate the point in the middle. Right? If something is very smooth, then that should be something I can take advantage of. And that is essentially the idea behind grappa. By the way, does anyone know what grappa is? Anyone here from Europe? Okay, it's an alcoholic drink, I think, in Italy or something. It's the name of a very famous drink. So one of the things about MRI is people are always trying to come up with the best acronyms. And so this is one of the better acronyms because it actually refers to an alcoholic drink. Okay. I think grappa probably related to some grades, right? So there's something there, but I don't remember exactly what the grapha stands for in terms of the acronym. But the idea is this, that I can acquire some calibration images which have all k-space, okay? And then I can sort of calculate, basically, let's say I'm missing this point in k-space. I can use neighboring points around k-space to calculate what that missing point is. Right? Because there's smoothness in my image. And the way I calculate those weights is I acquire a few images beforehand where I actually acquire all of k-space to figure out what those weights should be. And then I can, each image now is fully sampled, and then I can Fourier transform and Coil combine. So that's shown here with grapha. Essentially here, I'm going, with the grapha, I'm basically coming up with full Fourier estimates, okay? And then I can just do a sum of squares reconstruction. So this is an example of what these images look like. So for example, there are trade-offs when you go too fast. So here I've gone twice as fast, and look, the image looks pretty good. If I go 3 times as fast, I'm starting to see some artifacts here. And then here, 4 times as fast, clearly, I'm starting to see even more artifacts. Okay, so far what we've done is we've talked about parallel imaging, which is basically we're going to use coils, and we do the reconstruction, either image domain or the k-space domain. And in either case, we get a pretty good looking image. And plus we can go faster. Right? And so up until about 10 years ago, this was sort of the best you could do. Okay, so now we're going to talk about what has happened in the last 10 years. Okay. So here's the basic idea. So every image, you can represent it in a number of bits, right? So basically, you know, if you have a bit means, you know, how many values each voxel can take on, right? And so if I had a 16-bit image, then I could have the space of all possible images is about 1 billion if I have 120 by 100 image, because that's all the if you imagine all the values that all those images can take on. It's a very big space of possible images. If I have a 512 by 512 image, it's like 17 billion possible images. If I go up to a 32-bit image, now I've got 70 trillion or 1 quintillion images. So a huge space of possible images, right? Fourier transform, whether we use fully sampled or parallel imaging, tries to assume that I need to figure out any image in this whole space. Okay. And it will do its best to reconstruct that. So for example, even if the images don't look like what I want, it will still try to create that image for me. Okay. Because it's the same thing. It's like, if you don't know where you lost your keys, you have to like figure out every possible solution. And that's going to lead you to not only it's going to take longer, but it's not going to be optimal. Whereas if I say, well, you know, biological structures don't look like random noise, right? If I show you different images, you can say, well, yeah, that's biological and that's not, right? I mean, you can tell something biological from like a picture of the freeway looks very different from an image that you get from any biological structure, right? So your brain has already figured out, oh, this looks like a good image and this looks like a bad image, right? So the space of all sort of reasonable looking biological structures is much, much smaller than the space of all possible images. Right? Fourier transform doesn't take advantage of that, right? It has to search the whole space, right? And therefore it has to acquire a lot of data to actually come up with an estimate. So the idea behind compressed sensing and deep learning is, well, I just need to collect enough data, and then I can combine that with my prior knowledge to come up with a pretty good looking image. So I don't have to know everything about it, because there are certain options that can't be the right answer. So if I limit what can be the right answer, I should be able to go faster. So the first thing we can look at is compressed sensing. This came out of the statistics literature, and then by some people at Stanford and Berkeley, and then a grad student at Stanford sort of took it on and sort of applied to MRI, and that's really how that got into the MRI world. So here's the idea. Here's our famous Shep Logan image that some of you played around with your CD projects. here's its Fourier transform. And just let's assume we randomly downsample that Fourier transform. So we just like throw away like, I don't know, 70% of the data in Fourier domain. Okay, we're just gonna throw away the data. If I just do like a minimum norm estimate, like what we were talking about, this is my reconstruction. But if I apply a type of compressed sensing, in this case, it's called minimizing total variation, then I can end up with a much better estimate. And so the key is I have to put some prior assumption on what is an acceptable image. So this is the way we do it. This is just called data consistency. So this, for example, remember we said we could represent our data as S equals e to the m, right? Where this was the encoding matrix, this was the object, and this was the data I measure. And now I come up with an estimate, m hat, right? I want to estimate my object with some m hat. So e m hat minus s is just minimizing the error, right? I want whatever estimate I come up with, it should agree with the data, right? But if that's all I have, then I'm stuck with Fourier type methods. So what I do is I add an additional constraint. It's like, well, I also want to minimize something about my estimate. And typically, we're going to apply a transform to my estimate. And this is typically going to be a sparsifying estimate, such as a wavelet transform or a finite difference filter. So for example, if I have an image of just blood vessels, right? And if I imagine taking the derivative of that, then that's just going to have like edges at certain locations, but it's not going to have edges everywhere. And so, if I can make some assumptions about what the smoothness of my data is, then I should be able to use that estimate to control the possible reconstruction. So for example, here, let's say this is my k-space data, this is my object that I care about. If I do a transform here, a wavelet transform of this object, you sort of see most of the information is in certain parts of the wavelet domain and other parts are not. And anything that's noise will sort of be spread out everywhere. So I sort of just take whatever estimate I get that doesn't have too many coefficients in wavelet domain, it turns out I can get a pretty good estimate of my object. All right. So it works really well, for example, for things that tend to have sparsity in them anyway. So for example, if I look at an angiogram, this is what I get if I spend sort of what's called Nyquist sampling, which is what we've done in the class, where we acquire all of Fourier domain. And now here I've gone 10 times as fast. And you can sort of see here, I've done a pretty good job. Okay. Whereas if I just did low resolution to go faster, I'm not going to retain my detail. And if I just took my data and just did the linear reconstruction, like a minimum norm estimate, I'm also not going to get a very good estimate. So it's by imposing that constraint that the compressed sensing is able to sort of figure out how to do it. Here, once again, we sort of see if we do full sampling. Now we go 3 times as fast, and here we go 6 times as fast. So you can see it does remarkably well. Okay. But the problem with compressed sensing is the reconstruction takes a long time. And there are still sometimes artifacts that can't be explained. And whether or not an image should be smooth or satisfy these constraints everywhere, it's not clear because these are sort of man-made constraints we put on, right? And so lots of people will try different sorts of cost functions. Say, well, would I do this constraint or that constraint? What would I get? So this is just in the last few years, right? This paper was published in Nature in 2018. So just 3 or 4 years ago, deep learning has now come in and is already now available on clinical systems, which is remarkable. So basically a method that's, and if you think about, deep learning in terms of things like image, Jeff Hinton coming up with stuff, I mean, that wasn't that long ago, right? And so the idea is this, that if I take my data, and these are just different ways of collecting the data, I can go through my conventional reconstruction chain, which we've spent a lot of time in this course in, or I can just throw it into a deep learning network. Okay, in this case, this network has a few fully connected layers and then a bunch of what are called convolutional network layers. So it's not a deep learning course, but we're just going to give you some sort of insight into why this might work. Okay. And the idea is that it's going to take my data, right? And it's going to say, my data doesn't, like, here's my data, and what I want to reconstruct are some images that are like biological looking images, right? So those images lie in what's called low dimensional manifolds. Like if all the space is here, the solution space I care about is just some sort of subset of that. Right? So I'm going to look for those solutions that lie on that low dimensional subset. And the way I'm going to define that subset, that low dimensional manifold, is by training on a lot of data. Right, I'm going to train it on like the ImageNet data. And so it doesn't really, the remarkable thing is you don't have to even train this on MRI data, you can just train it on any image, any biological looking images, because biology tends to use the same patterns over and over again. So you can train on trees, plants, animals, stuff like that. And it does remarkably well. And even the implementation, I mean, this is from the paper, basically. That's all the code is, right? So now with things like TensorFlow and Keras, anyone can do this relatively quickly, right? And the results are pretty stunning. So for example, if I do, an MRI, we can do radial projections similar to CT. So if I did that and I look at the reference and I look at the best conventional image reconstruction, this deep learning does much better. Similarly, what's really impressive though is this one here. So, and then here is where I do undersample. So this is the full reference. Here I do compressed sensing. I do pretty good, but this automap does pretty much just as well, if not better. And here is pretty amazing. Here, I assume I misalign the Fourier data. I basically just like randomly shift the data all around the Fourier space. So things are just wrong, right? And this is what I get with conventional imaging reconstruction. Like, I just pass this through a Fourier transform like this. Remarkably, the automap can figure out, well, this is not a biological image. So it must come from some low dimensional manifold that has this image. So even if you put lots of mistakes in the acquisition, the deep learning network is smart enough to figure out, oh, actually, this is what the underlying image should have been. Okay. So an interesting thing is that, for example, so this is just an example. So sense is what we talked about. Okay, so here you can actually do sense in both directions, and it's just showing you that if this is what we really care about, this automap does actually better than the best sense recon. Okay. And so see, this is the error, about 10.3%. The automap does even better. It turns out that the basis functions that this network comes up with look remarkably like Fourier basis functions, right? You're sort of seeing these patterns. Okay, so on its own, it's figuring out what images should have in it. Okay, what are the basic building blocks of images? This is, let's see, just showing that in when you have very low SNR, when you have a lot of noise, automap can sort of say, well, okay, that noise is not, I can ignore that noise in creating my image, whereas a conventional image doesn't know what to do with that noise, okay? Because it's still within the bandwidth of what we care about. And so it knows how to get rid of that noise. Okay, so this is still an evolving area. People are still working on it, but there's different approaches. So for example, you can imagine taking your K-space data and doing inverse Fourier transform on it, get a very looking aliased image, pass it through a network and clean up the artifacts. So that might be one approach. Or you could take something where you go directly from the K-space data through your deep network and end up with your image. That's like the automap approach. You could do something where you have your K-space data and you stay in K-space, but deep learning cleans it up and then do the inverse Fourier transform. Or you can sort of do a hybrid approach where you sort of take some of the Fourier data and go right here and then clean some of it up, do an inverse transform, get some artifact image, pass it through a deep learning network, and you would get that image. So people are still experimenting with these different approaches. But clearly something like this is the most straightforward. And the advantage of it is that if you look at conventional imaging, it has a longer imaging time. The reconstruction is pretty fast because it was Fourier based. We can calculate that pretty fast. If I go to compressed sensing, I can speed up my images, but the reconstruction takes forever. Because you're trying to solve these convex problems and you're sort of searching around for these solutions. Machine learning is pretty cool because you can go faster. Training takes a lot of time, right? But once you have the weights, it's really fast to compute. So you can do the training offline, store the weights, put them on any scanner, and then the reconstruction is basically like almost instantaneous. You just multiplying by weights and then some nonlinearities. So it's very fast to do that. So basically, because what you've done with the training is you said, okay, I've defined my manifold, so now all I need to do is whatever comes in, the weights are going to drive the solution to lie on that manifold. Okay. So just to sum up a little bit, what we're doing here is in MR, we have some object we care about. We have some encoding matrix or forward operator, and we get our case-based data. Okay. If I don't have any prior information, then I have infinite many solutions. So all of these are okay solutions, right? But clearly, you don't have to be a radiologist to tell that some of these solutions do not look like a brain, right? So the question is, can the deep learning network learn the image manifold so it can say, well, this alias image is not a correct answer. And what I want to be able to do is take my deep learning network and go from here to something like that. Okay. And so that's shown here, where we're basically trying to project our data onto some low dimensional manifold that sort of captures sort of where we think images should lie. And so this still applies not only to MR, but also to CT. Basically, any sort of imaging modality now is going to have deep learning in it as we move into the future. Okay. So here's just a few examples of how it's being used. I think this is from probably a GE paper where essentially you can sort of see that. Let's see, so typically, if you do, we talked a little bit about Gibbs ringing. So typically, if you don't acquire enough data, you can sort of see this ringing here. Okay. And one of the ways we get around it is by apodizing the data. So we basically cut off window the data a little bit in Fourier space. You get rid of the ringing, but you also see that the image is much less sharp. Okay. So deep learning has been used to, for example, get it so that here we're sort of making the voxel size bigger and bigger, so from 0.2 up to 0.78. And you can sort of see here with conventional imaging, things start getting blurry. Okay. With this machine learning, it actually keeps things pretty sharp, even as you're increasing that voxel size, because it sort of knows what it's looking for. Here is basically using deep learning to go faster. So this is some images required with standard Recon. And in this case, we've gone, this has gone from 127 seconds to 6, so basically a factor of 2. And sort of still see it looks really good. And this is 15 to 11 seconds. And so basically, just by using these deep learning networks, you can actually go faster, and arguably the image actually looks even better. Okay. This is from, I think, Siemens, which is basically showing, for example, you can use it either keep the scan time the same and get sort of sharper looking images, or you can use it to reduce your scan time. I think this is still the same scan time, so just sort of showing more detail here. And here you've actually been using it to reduce the scan time. So this is like 3-minute scan time, and then they just divided the image, and this is the 2-minute scan time. And so it turns out that, you know, basically you're able to sort of, so you're not seeing like factors of 10 gain yet, but you are seeing the manufacturers introduce things where there is, you know, some substantial gains in scan time. Okay, so let's see. So are there any questions on that? So for problem 2 in the homework, just once again, you're just be experimenting with this coil profiles. Okay. If there's no questions, then let me just go through the part on diffusion MRI. So we're going to start it today, and then we're going to end it. We'll do a little more on Wednesday. This is probably one of the more important variants of MRI that's being used both in research and clinically. So I think it's good to sort of give you a sense of this, because the likelihood that you will have this scan done sometime in your life is pretty high on yourself or someone you know. So it's sort of good to know what these different scans do. So what we're going to do is we're going to switch gears a little bit from, so far we've assumed that the spins are just where they are, right? And we're going to make an image of the object, right? Now, what makes biology interesting, right, is things are moving around, right? So you have blood flowing, you have water diffusing. So when things are alive, there's a lot of motion. Right? And so what we want to do is start saying, well, what can MRI do to tell us what's going on with those moving spins? And so the first thing we're going to look at is diffusion. Okay? So diffusion, as you know, is just a random motion of, let's say, water, okay, or any sort of particle. Okay. So the classic example is, for example, if I had a bottle of perfume and it's all concentrated here in some bottle, I open it, then it's going to go from high concentration to low. You know, it's going to preferably try to equalize the concentration of perfume in this room, and so it will diffuse out, and randomly it will get to you, and you will start to smell it. So that's diffusion. And it sort of follows, if it's free diffusion, it follows sort of a Gaussian distribution. So for example, first is very concentrated, and then that Gaussian just gets bigger and bigger and bigger. And the width of that Gaussian, or how it's distributed, is related to D, is how diffuse the object is, the environment is. So the more I'm able to diffuse, then the wider, the more quickly it can spread. And obviously, the longer I wait, the more it will spread out. And so, for example, we can look at sort of diffusion either spreading out in 2D or like a 3D ball that's expanding out. So we want to relate it a little bit to MR. So today we're just going to give you sort of a sense of how you can use MR to be sensitized to diffusion. And we have to go back to understanding how spins add up to give you a signal. So imagine I have two spins that are in phase, right? And so if I look at a signal, they would add up together and I get some value out. Now what I do is I apply a gradient, okay, to these two spins. And so right now they're in the same place in space, so they experience the same gradient. So they would process the same way. But if they're diffusing, one's going to go one way and one's going to go another way, right? They're just going to randomly move around. So they're actually going to move into different parts of the field. Okay. And so I apply that gradient for some amount of time, and then, whoops, went the wrong way. And then I apply another gradient that goes in the opposite direction for the same amount of time. So if the spins weren't stationary, if I applied a gradient one way, they would speed up, and if I apply a gradient the same amount of time the other way, they would slow down, and they would just end up back where they started from. Okay, so if these spins stayed here, these gradients would have no effect on their phase, right? Because it's, remember, if I apply a gradient for some amount of time and then do negative with that gradient the other amount of time, the net area is 0. So there'd be no effect on the phase. But if they're moving around, now they're actually experiencing different gradients, different fields. And so at the end of the experiment, they've actually each acquired some net phase. And because it's random, each of them has acquired slightly different net phase, right? So now when I add them up, the signal is going to be much more decreased. Now, I imagine millions of spins doing this. The more they diffuse around, or the longer I wait, the more they're going to get out of phase, and the more the signal is going to go down. So it turns out diffusion MRI is very sensitive to diffusion. So this is how diffusion pulse sequence looks like. You apply your 90-degree pulse, you apply gradient one way, you'll wait some amount of time for things to sort of move around, and then you'll refocus it here. So anything that didn't move, these two things balance out, right? So they stay in phase. But if they moved around, they're going to get out of phase. And that's going to reduce your MRI signal. This is just another picture. So for example, and we actually, the cool thing about MR is we actually have some control over how that signal goes down. For example, if I apply a weak gradient, okay, then as these move around, they get out of phase a little bit, right? Because I haven't really changed how much the field varies with space. But if I have a very strong gradient, right, where the field really varies a lot with space, then as they move around, they're experiencing very different parts of the field. So I can actually tune my experiment to be more sensitive to diffusion. So what that means is that the signal I measure is going to be proportional to the exponential of minus b times d, where d is the diffusivity. Okay, so the more diffuse something is, the quicker the signal is going to go down. Okay. B is something I control with the gradients. So the bigger I make those gradients, the bigger b is going to be. That's B is called the B factor. Okay. So I want to make that B factor big enough such that I get sort of interesting decreases in my signal. If I make it too big, then everything's going to cancel out, and then I'm not going to get a good image. So part of it is picking your B factor. Typically, people might pick B of like 500 or 1,000 is typical. So what I can do, the other thing I can do with B factor is, since this, we know that this is proportional to exponential minus B times the diffusion factor, and here ADC stands for apparent diffusion coefficient, I can also map my signal at different B values. And if I take the log of both sides, I should get a linear relationship of this. And so the slope of this line, as a function of B factor, is my apparent diffusion coefficient. So I can actually measure what the diffusion coefficient is. And so the question is, why would I care about this? Like, where is this useful? So here is a T2 weighted image, okay, of a person. Now, if you're not a radiologist, I mean, you might say this looks pretty normal, right? I mean, maybe you could argue that something's wrong in certain places, but if you just had like a second to look at that, you'd be like, oh, okay, that looks pretty normal. Yeah. There's nothing really drastically different that pops out at you. Maybe something's slightly different here, but you know, is it really different? It's hard to say, right? If I do a diffusion weighted image of this person, notice this. Clearly, you don't have to be a radiologist to say, well, the left and right side look very different. Yeah. So what's going on in this person? If I do an ADC map, which is mapping the diffusion, you notice the diffusion in this area is dark compared to the diffusion on the opposite side. So that means for whatever reason, in this part of the brain, there's less diffusion of water, and therefore this MR signal is brighter. So this is the basis of diffusion imaging, and one of the biggest applications is applying it to looking for stroke. Okay? So it's still a very standard method. If someone has had a stroke, it turns out that in many cases the diffusion of water goes down. Why that happens is still not known for sure, but when the cells die, the diffusion of water, if there's, for example, active transport of water, then that transport would be decreased. Maybe the cells are swelling, that's reducing diffusion. But for whatever reason, empirically, people find that diffusion of water just decreases wherever there is cell death. Here's another example. Here, once again, here's a T2 weighted image. I mean, there's definitely something going on here, right? I mean, these parts look slightly different. Maybe there's more fluid here, right? Because it's a little brighter on the T2 weighted image. But, you know, unless you're an expert in this area, it's hard to say, is this part bad or is this part bad? Like, which part is normal, right? it's very obvious if you look at diffusion weighted that this whole part has remarkably lower diffusion than the rest of the brain. And then if you look at an angiogram, which measures where the vessels are, you can sort of see in this person, this vessel is obviously pinched here. Okay? So this whole area is not getting any blood flow and basically died. Now, whether that can be recovered or not is an area that people are still trying to figure out. Like, okay, if you have a stroke, does it make sense to try to open up the vessel and try to get blood back to that area? Can you revive the area, or is it just dead and you have more knowledge? So that's still an area of research. Right? Okay, so I think that's it. We'll end a few minutes early today. On Wednesday, we'll talk about how we use diffusion imaging to map the wiring in the brain. And then we'll also look at how we use MRI to look at blood flow and blood velocities and sort of more interesting, more dynamic applications of MRI. Okay. So we'll end for today. And if you have any questions, I'll be around.